We have our afternoon session, Transforming Translational Sciences from Biomarkers to New Approach Methodologies. I would like to take this opportunity to welcome my colleagues, Nicholas King and Graham Marsh from our Predictive Safety Testing Consortium, and they will kick off our session. Thank you.
Thank you. So I'd like to kick off the session with a brief overview. Jeff Siegel in the opening session this morning stole a lot of what I was going to talk about, so I'll just reiterate that with some slides. So two of the areas where Jeff highlighted the benefit of PPPs for advancing biomarkers in translational sciences. The first is using biomarker repositories as a utility for identifying biomarkers for efficacy or prognostic or safety. And so the work that we've been doing at CPath, well, that Nick has been doing really most of the work here, is bringing together kidney biomarker data into this biomarker data repository. And this is intended to be a cohort of kidney data that can show normal ranges for kidney safety biomarkers, and then hopefully that will translate to the development of new biomarkers in the future. The other area where we have been working is in NAMs and trying to move them towards a qualification for regulatory applications. And so just briefly, if you're not familiar, new alternative methods or new approach methodologies are in vitro tools that will replace or supplement animal models. And the two on the top, the complex in vitro models, that's the organ-on-chip or organoids or 3D tissue models, is what I'm going to focus on, but I think also Tina will probably speak to the in silico models or the AI tools that we could be using. And I want to highlight that these are human relevant models, so they are more predictive. They can be much faster than animal testing. And as an added benefit, we can reduce the animal burden and replace some animal usage in science. And so what we have done at PSTC, last year in September, we hosted a workshop to bring together regulators, academics, model developers, and pharma industry scientists to discuss the qualification of liver models for regulatory applications. And the QR code here will bring you to the CPath website that has recordings, slides, and a white paper that summarizes the discussion at the workshop. And the takeaway from all this was we think that we can bring complex in vitro models out of the early drug discovery space and have them be useful for regulatory decision making. And so this is the ISTAN qualification pathway. And the point I want to highlight here is that complex in vitro models have been widely used for drug discovery, for identification of molecules, for optimization of molecules, but their applications for regulatory sciences have really been lagging behind. And the two ways that we want to see complex in vitro model data included in regulatory submissions is either sort of ad hoc as supporting data as weight of evidence for showing the safety or efficacy of a therapy, or ideally qualifying these tools as drug development tools through the ISTAN program. And so in the September 2023 workshop, we brought together all of these stakeholders for a discussion around context of uses in the drug-induced liver injury safety space, and really looked to identify areas where we could standardize a framework to support FDA qualifications. And so we had a great panel of experts giving talks. As said, all this material is on our website, so please look at PSTC on CPath, and you can see all these talks and slide decks. It's a very useful resource. But David and Tina were both present to give their opinions on applications for complex in vitro models and building credibility assessment frameworks. And so the scope of the project, and so from your left, my right, discovery, target, hit ID, lead ID. This could be a whole other talk about how these models are validated and build confidence inside of companies as they're developing drugs. But what we really focused on was the regulatory science space and where these models might be included in INDs or NDAs to support a drug moving forward into the clinic. And the goal here is to establish evidentiary considerations for the analytical performance and biological characterization to build confidence that these models are predictive and show the right biology. And so, I mean, not to get into the weeds here, but we were focused on liver models. And so liver should produce albumin and urea. It should have enzymes for metabolizing drugs. It should have transporters. And so as you're building these models, as you characterize them, and the white paper goes into this in much more detail, you can think about what sort of qualifications for biological or for analytical performance for baseline characterization are necessary to build competence in these systems. And likewise, for the biological performance, the question is, do these models respond the way you think they should when given an insult, given a drug? And because these models are, in a dish, they're very accessible, you have a lot of options for what assays you run as your endpoints. And translating those in a dish endpoints to clinical endpoints is a challenge. It's not straightforward. And so considerations for what markers you're going to use become very important. And so, for example, looking at ALT or AST, these are clinical markers that are commonly used for assessing liver function. And so, that's a recommended assay that you would run to build confidence that your system is responding to a known drug that causes liver injury. And so Jeff mentioned the emulate context of use, the comparing a known toxicant to a novel drug to de-risk it. That's a prospective safety indication. We also talked through some other ones as well. Potential context of use statement here is using a liver model as a tool for retrospective toxicological analysis if DILI is observed in a clinical trial, to give clinicians a tool that they could use to assess the mechanism of injury of a drug and determine whether or not that drug is safe to continue in a clinical trial. And of course, for the specific context of use, I don't have a context of use t-shirt here, but this is exactly where you would want that. The specific context of use is going to drive what your analytical and biological considerations would be for characterizing that system. And so we're stuck in this sort of, right now, regulators, when you talk to pharm tox reviewers, they say that they need more complex in vitro model data to be comfortable making decisions about the existing models. And developers need guidance on what the guidelines should be for making these models. And what the developers make determines what the pharma users can actually test their drugs in and submit to the FDA. And then that leads back to the regulators again. So in order to break the cycle, we think that these meetings, these face-to-face, these facilitated discussions are important for driving the field forward and actually moving these tools from current use to actual regulatory decision-making. And so, yes, it's the afternoon on the last day of this meeting, and I'm sure all of you are sad that this is ending, but I have good news. We will be back in 2 weeks to discuss complex in vitro models just up the road in Bethesda. Please do join if you're interested. It's a public workshop, so we will be discussing more evidentiary considerations for these models. We will be trying to advance the field. We have a wonderful panel of experts lined up for speaking, and so please do scan this QR code and register if you're interested. And so with that, I believe I can introduce the panel, and thank you.
Great. Yeah. Thanks, Graham. Yeah, so thank you, everyone. So first, what we'd like to do is I'd like to give our panelists just a brief opportunity to go through a little bit of their perspective on applications of novel approach methodologies and where they're approaching this from their background. And so first, I'd like to lead off with Dr. Dan Tagley from NIH, where he works within NCATS. So Dan, if you'd like to take a moment.
Thank you, Nick. Good afternoon, everyone. As Nick said, I'm Dan Tagley. I'm the Director for the Office of Special Initiatives at the National Center for Advancing Translational Sciences at the NIH, and have been leading the effort within the NIH over the last 10 years or so in developing complex in vitro models, also known as microphysiological systems, tissue chips, or organs on chips. We've had, over the past 10 years, worked through demonstrating and developing the technology towards safety and efficacy assessment for disease modeling and use for efficacy, as well as building confidence through our microphysiological systems database, and also through independent testing centers that would seek to replicate the robustness and the performance of these complex in vitro models. The latest iteration that we have going from efficacy is clinical trials on chips. That is an ongoing program. And then we also have just recently launched, and Jonathan is one of the main PIs on our translational centers for MPS. So these are activities that are partnered with FDA as well as critical path in terms of defining specific context of uses for complex in vitro models and how they can be qualified as drug development tools. Coming off of that would be a larger NIH-wide effort beyond NCATS that involves all of the NIH, what we call the NIH Common Fund, which is to develop a program called Complementary, which stands for Complement to Animal Research and Experimentation. This is essentially to develop new NAMs, either solely or in combination. So for example, CIVMs with in silico approaches called combinatorial NAMs, essentially to augment some gaps and deficiencies. But really the point is to be able to position this new NAM approaches towards areas of unmet needs where in vivo models don't exist or have consistently performed poorly in various assays. So this is a new program, excuse me, that will be launched in about a month or so. And it will be a 10-year program that will seek to develop through our technology development centers. There'll be centers set up across the United States to develop the technology in terms of combinatorial NAMs. We will have a central database that will take in both preclinical and clinical data, as well as in silico approaches, in chemical approaches, and in vitro approaches. And then lastly, the major component of this, as we heard from Klaus earlier this morning, is the need for global public-private partnerships. So a component of complementary is to establish a validation and qualification network. And so we're in the process of discussing this framework, this validation qualification network, not only with FDA, with CPATH, with EPA, USDA, and other regulatory agencies, consumer safety, but also having conversations across the world with EMA, ECHA, and EFSA, and also with Australia and Japan and South Korea, so that we can quickly adapt the technology and have global harmonization in terms of regulatory acceptance. And a key component also complementary is to not only bring it to regulatory acceptance, but also to train new scientists, next generation scientists, as well as workforce development to make sure that this new tools, as Klaus mentioned this morning, a solution is not a solution unless it's adopted. And so our effort with the training, outreach, and workforce development is to make sure that these tools are being disseminated and widely used. And so with that, I'll turn it over to Madhu.
Thank you, Dan. My name is Madhu Lalnag. I'm the Chief Scientific Officer of a company called InSphero. We're based out of Switzerland. And for the last 14 years, we've been developing complex 3D in vitro models, which are human-centric for safety and efficacy. The perspective I'm bringing today is actually twofold. One is as a CSO for a company that is bringing the developer perspective. You heard beautifully from Graham that there's this cyclical relationship between the regulators, the developers, and pharma, right? With everyone sort of having a dependent relationship on what the other party is bringing to the table. And we're here today having a conversation, and I really do believe that these conversations are moving the field forward. We are gaining from this. The other perspective that I'm bringing is as a scientist. I spent 10 years in academia at NCATS, actually on the preclinical side, developing 3D models for oncology. We ran some of the first 3D oncology models for high throughput screening. And I bring that DIY approach, comparing it to the standardized approach that developers now offer the industry, to really do a compare and contrast, to be able to allow people to understand what it is that they're able to benefit from adopting standardized models, what information it's going to give them and what information it's not going to give them. It's equally important to sort of know both sides of the coin. And with that, I'll turn it over to Tina.
Hi, good afternoon, everyone. This is an audience I'm not supremely familiar with. As my background, I'm a mechanical engineer, and I spent 12 of my last 16 years at FDA in the medical devices space. My background from a regulatory perspective is cardiovascular implants. I spent 7 years as a regulator, both as a reviewer of pre-market applications for cardiovascular implants, supported other regulators by serving as an expert consultant on computational modeling approaches and in vitro testing. And I went into the Office of Science and Engineering laboratories and spent 5 years running a group where our focus was try to develop frameworks, standards, guidance documents to support the use of computational modeling in the regulatory review of medical devices. I spent a large part of my time in CDRH trying to understand what the needs were, not just for the regulators, but also for the industry. And we ended up developing what's become a pretty popular standard through the American Society for Mechanical Engineers, is what folks have been calling the VNV40 framework. And VNV40 just stands for the name of the subcommittee that developed this framework. But effectively, it's what we were calling a risk-informed credibility assessment framework. We really pushed the definition of context of use to really drive the evidentiary bar for evaluating and validating computational approaches. We started a large-scale project in the Office of Science and Engineering labs that just ended this summer, which was towards the development of in silico clinical trials and developing a specific credibility assessment framework that allowed companies to be able to harness patient-specific models, building population-specific models to complement or support enriching clinical studies for medical device development. This hierarchical analysis and framework just was published a couple weeks ago. There are 2 papers out of CDRH, and why I'm here is I've been engaging with the Center for Drug Evaluation Research over the last few years, trying to find ways to translate that framework into approaches for drug development. I have been working with the leaders of the Quantitative Medicine Center of Excellence. We wrote a couple papers thinking about how can we apply the same concepts to drug development models used in drug development. So that's the expertise and perspective that I get to bring to this community. So pardon me if I really don't understand the intricacies of drug development or of the biologically relevant models. As an engineer, we did focus mainly on the physical structures of devices. So that piece is something that I'm learning, but I'm excited to be here and participate in the discussion. Thank you.
Thanks, Jonathan.
Good afternoon. Really a pleasure to be here. My name is Jonathan Himmelfarb. I'm a nephrologist. I recently moved to the Mount Sinai School of Medicine to co-direct a new Center for Kidney Disease Innovation. I came from the University of Washington, where for 15 years I was a professor of medicine, pharmaceutics, and bioengineering, and where I founded a Kidney Research Institute and then a Center for Dialysis Innovation. My connection to this field really stems from programs that Dan really pioneered and led through NCATS, and I've led a group of really talented, diverse academic investigators who are pharmacologists, toxicologists, and bioengineers over the last decade to develop these complex in vitro models related to kidney diseases. And we've been funded through that time via NCATS, also the Center for Advancement of Science in Space and NASA to study space biology and also accelerated aging in these systems as well. And we were fortunate enough to be selected to be part of the new TRACE MPS consortium, funded again by NCATS. And in that, we have proposed 5 different contexts of use for drug development tools using our complex in vitro models, 2 on the tox side, 2 on the drug discovery side, and 1 on the PBPK side. So I'm coming from the academic developer perspective to this meeting.
Thanks, Jonathan. Hey, David.
David Strauss, it's a pleasure to be here talking science. For the past 5 months, I've been the acting chief scientist at FDA, but we're implementing a very large reorganization on October 1st, and that affects 8,000 of the 18,000 people at FDA and brings 1,000 people into the office of the chief scientist. So it's like just management all day, reorganization. This room reminds me of the, even though we were in Bethesda, the workshop we had that was described earlier in the talk last September on complex in vitro models and how to bring them forward into regulatory use. I'll say, as my background, I've been at the agency for 14 years, 5 years in the Center for Devices and 8 years in the Center for Drugs, where I led the Division of Applied Regulatory Science. And that I don't know, the title of this session is sort of in line with the mission of what we were doing there. I would say some of the thoughts I shared at the meeting last September, it was last September, yeah, a year ago, was, yeah, I think the science is here in many places, and it is, you know, the science may not be here to replace all animal studies, but we can make inroads in specific areas, and it's about getting the stakeholders together to figure out what are the areas we can address. I mean, at FDA, we can't, you know, unless Congress passes a law and says, you know, every drug company, you know, is not allowed to use animal studies at all. And I don't think that would be a smart thing for public health. You know, we can't force companies to do things. And so the developers of these technologies need to work with the pharmaceutical industry scientists, and then with the regulators as well, and figure out where are the points, the questions of interest. That's something that I know Tina described at the workshop last year. And then how do you go from that question of interest that you're trying to solve, whether it's in safety or effectiveness, and define a context of use of where you can use a new technology. And we have had examples in real drug approvals where we've used alternative methods. And I've talked about some of those previously. We had a FDA science board meeting 2.5 years ago. I led an internal group to then go to the science board, and we charged a subcommittee to the FDA science board. If you're not familiar, the FDA science board is an advisory committee to the commissioner on cross-cutting issues that the chief scientist office oversees. And we actually are going to have it's been announced in the Federal Register, there's going to be a science board meeting October 7th, I believe. It'll be remote, and that subcommittee will actually give a readout of their report. These are people outside the agency, of their recommendations to FDA on related to alternative methods and implementing them at the agency. So yeah, excited to be here with different colleagues, past and present. And Tina and I were actually in the same division in the Center for Devices in the first couple of years at FDA, and now we're in the same office again. So yeah, looking forward to further discussion, maybe talking about some examples.
Yeah, no, thank you everyone for this wonderful introduction. I really like having a diverse group of scientists up here to talk about how translational sciences and utilizing new approach methodologies are going to have an impact in drug development over the next 5 and 10 years. And so kind of as a first question that I'd like everyone to kind of discuss is, one, first, where have you seen NAMs positively impact drug development to date right now? If you've got some specific examples that you'd like to bring up. David, you mentioned a few that you've talked about in the past. You want to give a moment for maybe one of those.
Yeah, so I will, okay, I'll talk about 2 different examples I'm familiar with that are, yeah, the reviews are, I can think of 3 actually, and there's actually links in the science board presentation from 2.5 years ago. You can go see the reviews. The first one was related to, I'll say, alternative methods to assess effectiveness of drugs for a rare disease. And there the situation was The first type of review we applied this way of thinking and methodology in was for cystic fibrosis. And there was a cystic fibrosis drug that had been approved to treat, I think, 11 genetic variants, specific genetic variants. But there's actually thousands of possible genetic variants for cystic fibrosis and like many genetic diseases and rare disease drug development. I wasn't here for the other, I guess, the focus yesterday, but it's challenging. There aren't many patients. It's hard to run clinical trials. And so how do you, if you have a drug or some other therapy that may work for certain genetic variants and not others that are all classified as cystic fibrosis, how do you determine if the drug's going to be effective? And in that case, the sponsor came up with an in vitro model, a cell-based assay that would express the genetic variants in the cell-based model. They were able to correlate the results of the in vitro assay with the clinical data that did exist. And then there was a very thorough review that occurred, but the agency did expand the approval for that drug to genetic variants where there were few or almost no patients with that specific genetic variant in the clinical trials. And that was done for a Fabry's disease drug, a similar type of thinking and approach. So that's actually like an alternative and using an in vitro model for replacing when you can't do a clinical study. Another example where it was, there was an option could have been to run an animal study was for the Department of Defense was supporting and wanted the development of a high dose naloxone auto-injector. It was approved for possible, the indications for use says something along the lines of can be used if there's chemical warfare for in the military or for responders for incidents if like fentanyl or carfentanil was used in a not good way. Yeah, it's hard to run a clinical trial there. An option could be to run animal studies to demonstrate effectiveness, and there is this animal rule-based approach to approving drugs, but there are limitations to animal models, and the approach that was used was to use a quantitative systems pharmacology model, and the evidence of effectiveness was really based on an in silico approach for that indication. So those are a couple of real examples, and there are other safety areas as well.
Great, thanks, David. Dan or Madhu, did you have any examples that you wanted to kind of?
Yeah, so certainly along the lines of what David had said, I've seen instances where there are clear models that are lacking in rare disease populations, or even in understudied populations, like I think this morning we were talking about neonates. And so we have, for example, tissue chips that's looking at preterm labor and modeling maternal fetal interface. We have examples that have actually gone as supplementary data to an IND submission to the FDA on looking at polyneuropathies where no animal model has existed before. So those are occasions where efficacy data can be evaluated using NAMs. A second area that we have seen certainly at NCATS would be in the area of precision medicine, being able to capture electronic health records of many individuals, create digital twins of every individual, and then be able to validate that with CIVMs. Are there computer predictions, borne out when you test them out on in vitro platforms? And I think the third area, and David touched on this, is I've seen it be deployed in the context of national health emergencies. We've gone through from the opioid pandemic or opioid crisis to the COVID pandemic, and time again, we have seen human relevant CIVMs being used to properly predict candidate antivirals or new pain meds that are more amenable analgesics in the context of, I think you mentioned even naloxone. So we have a tissue chip that would actually model efficiently the respiratory arrest because of the boxy neurons found in the lungs, which is not adequately addressed in any in vivo model systems. So those are now in the process of being developed, and some of them have gone on to the clinical trials. And so there's been a number of successes in the use of NAMs being deployed both in preclinical as well as clinical.
Great, thanks, Dan. And
Yeah,
did you?
sure. I can, I'm loving that we've talked about genetic polymorphisms, with both David and with Dan, because that's something we definitely see, with liver disease. When you have different populations with single nucleotide polymorphisms, the donor differences that you can see in a human relevant 3D in vitro model that you're able to test for efficacy. We already have a customer that's been able to take this into phase 1, phase 2 right now, phase 2A. And this is sort of, this is a disease for which there is no animal model that exists. So it's really very satisfying to see this sort of move forward. I think there's another area that David touched upon that I'd like to expand upon. I'm trained as an oncologist, and I know that we do not like to give up our animal studies. We don't like giving up in vivo pharmacology studies. But I will say one area where NAMs have really helped oncologists has been to de-risk how many animal studies you do. So it's not always about giving up all the animal studies and replacing all of them, but to really de-risk what animal studies, which animal study you really want to prioritize. So you use your NAMs to de-risk that and then go after the one that you want. So that's another area that I think that we've really made headway in.
Great, thanks. Any other examples, Tina or Jonathan, you'd like to raise that you've have experience with?
So I could give some examples from our own work that are not, that at this point in time haven't impacted the approval or the regulatory process, but they show the power of these kinds of systems to understand disease mechanism in a way that can contribute to both the drug discovery process and also understanding nephrotoxicity. So I was thinking just a moment ago, there are about 5 different examples from the last decade of work that we've done. So one, yesterday there was a discussion about autosomal dominant polycystic kidney disease. And our group just recently, a couple months ago, published a paper that really is a precision medicine concept paper for therapeutics, really a unique paper for autosomal dominant polycystic kidney disease. Polycystic kidney disease results from mutations in one of 2 different genes that were each cloned about 25 years ago, polycystin 1, polycystin 2. Polycystin 1 looks like an orphan G-coupled protein receptor. Polycystin 2 is a cationic channel. We know they form a complex together in the cilia, and somehow that results in cystogenesis. In particular, PKD1, the gene is a very big gene, and there are hundreds of different mutations that run in families. And this is a disease that's autosomal dominant, where you see generation after generation end up on dialysis with end-stage kidney disease. So there is a group, about 40% of the mutations are truncating mutations, which basically stop transcription of the entire gene. So you get a very truncated gene. And it's known that phenotypically, these are the worst mutations. It results in end-stage renal disease at a much younger age than some of the other mutations. They're base pair mutations, and there are no animal model homologs for the human mutations. So using kidney organoids and base editing techniques, we recreated in kidney organoids these base mutations that lead to a worse phenotype. And we showed that a class of drugs called read-through therapeutics, which were originally developed for muscular dystrophy and cystic fibrosis, can reduce cystogenesis in a model. We also have made models using CRISPR where we knock out the PKD genes, et cetera. So different cystogenesis phenotypic models. And we showed dose-dependent effects of these read-through therapeutics only in the mutations that are truncating mutations. Other mutations that cause polycystic kidney disease no biologic effect. So it's starting to bring precision medicine to polycystic kidney disease to say not everybody with a mutation in this gene should be treated with the same family of therapeutics. So it's an example of how we can learn something about both precision medicine and mechanisms for future therapies. That's one example. Another interesting one is with SARS-CoV-2. That hit all of the world incredibly. And not long after that, the Penninger group showed that ACE2 was the major receptor for SARS-CoV-2 in the human body, certainly in epithelial cells. But there was some controversy about that. Was it the only receptor? And there were co-receptors. And many in the early days of SARS-CoV-2, many people developed acute kidney injury who were critically ill with SARS-CoV-2. And the question was, is this due to direct infection of the kidney, or is this just due to systemic illness? So again, we took kidney organoids, and we, using again CRISPR, knocked out the ACE2 receptor, which is very prominent in tubular epithelial cells. And we transfected with live SARS-CoV-2 fluorescinated virus and showed, yes, the proximal tubule cell, which is rich in ACE2, gets infected. But if we knock out the ACE2 receptor, there's absolutely no infection. We also were doing a clinical study at that time in the ICU funded by the CDC, collecting urine and blood samples from people with respiratory failure in the ICU with and without SARS-CoV-2 as the etiology. We did a proteomic study in the urine and identified 9 proteins that were differentially expressed in people who had respiratory failure due to SARS-CoV-2 versus NH flu or any other bacterial pneumonia. And when we looked at our organoids, we found that 8 of those 9 proteins were transcriptionally upregulated after exposure to SARS-CoV-2 virus. Then we tested a vaccine candidate that was developed through the Institute of Protein Design at the University of Washington that has picomolar efficacy at binding the spike protein, showed dose-dependent effects at preventing kidney infection. And we submitted that paper for publication in September of 21. So about 18 months after this. It shows how fast you can move and learn something about disease pathogenesis using these in vitro systems.
That's great. Thank you, Jonathan.
Yeah, and then we've done others for sake of time. Maybe I'll move on the tox side as well, where we've made mechanistic insights that not all nephrotoxins are nephrotoxic by the same mechanisms.
Oh, excellent, excellent. And kind of moving on to a little bit of in terms of looking at adoption and applications in the future. I mean, I think a first component is really is looking at having confidence. And so Tina, with your prior work in developing credibility assessments and frameworks, what do you see as key components from what you've been looking at so far to bring into for some of the NAMs in terms of building confidence in the models?
Yeah, thanks. I have a couple of remarks on that point. Before I jump into that, I did want to just emphasize something that Dan mentioned about the complementary program, which only struck me recently, despite how many times I've heard you talk about it, which is that the focus of the complementary program will be on human relevant models. So the emphasis not on developing NAMs specifically for animal models, but for human relevant models. And there's a couple interesting challenges we can go with that conversation. One is, okay, if we're going to be focusing on human-based models, how do we get sufficient data to be able to validate those models? And some of the conversation is, well, let's compile data, let's share data that already exists. And if you're someone who has developed any kind of model that you try to validate, if you take data from a source and those data were explicitly collected for your validation study of this particular model, there can be challenges. So if you actually don't get to a place where you feel like your model is sufficiently valid, you can ask questions like, well, is it because my model isn't a good enough model? Or do I actually lack the data I need to do that validation? So one of the things I will get to be able to be part of the validation qualification network. And one of the things I hope to emphasize as that group moves forward is figuring out ways to combine resources to have targeted data collection specifically for the validation of those names, because while I think it sounds like a really nice idea to try to capitalize on data that we already have, since many of us feel like we're sitting on so much data that we can use, not being able to sit at the table to a priori discuss what are the pieces of the model that you need to validate and what are the data you need to validate that, to borrow from data that already exists can bring its own challenges. So I'm hopeful that part of the resources out of the complementary program will be to work with those mature human relevant NAMs and think about ways in which we can actually support the development of those NAMs by getting targeted studies to show that it's possible, so that when we don't achieve validation, it's not because we don't have sufficient data. So for me, that would be one piece I would add to the conversation. And just as some examples to maybe borrow also from the medical device space, because I do think there's some synergies in that. For those of you that aren't familiar, clinical trials done on medical devices are really small compared to drug studies. We're talking like a few hundred people, not a few thousand. And so the idea that we can build human relevant models to answer specific questions, not to replace the clinical trial, but to reduce the burden of the clinical study such that there are questions we can answer with these computational approaches, that has been demonstrated by two examples out of CDRH. There's a Bayesian framework that allows for that enrichment to happen. And then also a study that FDA conducted that showed that their computational model actually did better than the clinical study. And if that interests you, I'm happy following this discussion to meet you in the hallway and share those references for the sake of time.
Great, thanks,
Yeah.
Tina. You know, and everyone else, what other components do you see in terms of building, you know, credibility assessment and as we're working on a qualification framework, what additional components do you see as important right now?
Yeah, so over at NCATS, what we had done to build trust and confidence in, especially in CIBM or microphysiological systems, is to bring the major stakeholders, the FDA and pharma, into the discussion early on. And so we invited them to look at raw data through our microphysiological systems database. So even before it became public, they can look at the data, be able to compare that with existing legacy data from in vivo models, how they compare, and then be able to move forward. So we've seen members of, like we have a partnership with the IQ MPS affiliate, about 26 pharma companies who have looked into some of the data and have shown confidence enough in the technology that they have started using MPS for internal decision making. So that's one example, is just really working with the community and making sure that they have access to the data even before it becomes public, and to be able to be part of the process in terms of continuing to develop and the technology and demonstrating its utility. As Tina mentioned, you know, part of that is making sure that all major players in the ecosystem is also being engaged and involved. And so NAMS is not just unique to pharmacological development. We're seeing an increased use in biologics, but certainly in assessment of environmental toxins, in cosmetics, in industry, from chemical pesticides and herbicides, and looking at environmental toxins. All of this requires the use of NAMs, or at least the development of more sensitive human-based NAM technologies. And so I think as one sector of the industry gets more and more confidence in the field, others will follow suit and develop. And so that's what we're trying to create in the validation and qualification network, is sort of like a regulatory framework, not just for pharmacologics, but, you know, in terms of the whole use of NAM technology across many industry applications.
Great, thanks. And Madhu, you know, in InSphero, you know, in terms of, you know, building confidence for your models that you're developing, you know, what are some of the key points that you bring forward right now?
You know, I go back to that cyclical diagram that Graham presented. I think it's so important, you know, the qualification workshop. I think bringing everyone to the same table to talk about these things. I spent a short time at the FDA, and I think my perspective then was, we need people to share data in the pre-competitive space. And correct me if I'm wrong, you know, I still don't think people are sharing enough data. And I think till there's data being shared, there's that confidence from the developer's perspective, the confidence from the farmer perspective really isn't there that, you know, this is robust enough. This is giving us the answer to this particular question. And I think till we get there, there's still going to be that area of slight amorphousness that, you know, we don't have 100% confidence in this.
Great, thanks. And I think that kind of leads into the next area I wanted to bring up is, you know, what do you see? I mean, so you brought up one of the hurdles for overall adoption, but where do you see everyone see a few opportunities to foster adoption in drug development for regulatory decision making right now with the use of NAMs?
Well, I'll add I'll put a plug in for so Tina spoke about it some, the coming up with robust credibility frameworks and how you look at the truth, the question you're trying to answer, and defining it is very important, and I think is the hardest part often. There's uncertainty in every model, whether it's a clinical trial, which is a model for, you know, a patient in the real world that may take a drug, and yeah, how you, one thing that resonated with me, I remember from the discussions at the workshop a year ago, is you're usually going to have multiple pieces of information when you're a regulator and you're receiving data from, well, whatever kind of data, non-clinical, clinical data. And so in determining what is an acceptable level of uncertainty or predictive ability of a given test, you need to take into account what other information is present. What's the risk if you get it wrong versus not? And there are some good frameworks. I think that VNV40 that was developed on the device side is very useful, and the Center for Drugs has been working to adopt it in certain areas. I think that, yeah, the defining truth is very challenging. And I had the opportunity to work for about 10 years on this cardiac safety initiative on the drug side. It was called the SIPA initiative. And it was the goal of taking in vitro and in silico technologies and using them to assess risk of drug-induced arrhythmia. And yeah, I think in the development side, yeah, one of the most challenging aspects was defining the list of like 28 drugs that had different gradations of risk that had been on the market for a while. That's very important. And people always wanted more drugs, and that list isn't big enough. But I don't necessarily agree with that. I mean, if you're developing an AI model and you need or some other type of learning, black box learning model, you may need you do need lots of data with an answer, but that can be tricky. And so I think the credibility framework, considering what is the question you're addressing, and bringing the right stakeholders to consider, getting the regulators in the room with the technology developers, the drug development tool, or other FDA regulated product area. It was referenced, you know, Dan was mentioning cosmetics or foods, or now I'm in the realm of overseeing cosmetics. It's actually moving into the office of the chief scientist. So we've been all about cosmetics recently, but yeah, defining the truth and you know, getting the stakeholders together again, I think is very important.
Great.
Can I
Go ahead,
build on that? Because there was two things I wanted to make sure I shared with this audience today. The other piece for me, not being a regulator in drug development. So my colleagues, I apologize, I'm going to say something that might seem like, shut her up. But when I think about the fact that there's a validation approach that came out from ICVAM recently, and I know that might be one of the driving documents to start the discussion in the VQN. There are aspects around defining the context of use, again, which is key, biological relevance, technical characterization, data integrity, this independent review, which is also very interesting. In part of this discussion of these topics, is this comparison to what already exists. And I would really like to challenge CPATH as you guys bring folks together, especially at the meeting in a couple of weeks, and thinking about how can this be a conversation to say, when is it really relevant and necessary for us to do that? And the reason I say that is coming from the space of computational modeling, we've had to spend so much energy showcasing that our models can replicate what's done on the bench, when in reality, that's not what the model is for. The model isn't to replicate what's on the bench or, pardon me, what's in the animal model. If we're really focused on human relevant models, then that model is really developed to do something else. So comparing it to the gold standard today may just actually be a lot of resources spent where not a lot of new learnings are there. So it's going to be really critical in that conversation of saying, here's this model that's on the table. As we're talking about the context of use and the risk of using that model, it's equally important to not just focus on what you think could be wrong with the model, but what can we learn from that model that we can't learn now with the other models we have? And how do we give credit to that side of the conversation of that model, in addition to, well, we might not be sure about its biological relevance in this case, but we're really confident here. So looking at the NAMs as what can we get from those, not just to say, let's compare it to what we're already doing and check the box that we feel good about. And I know that's because of the uncertainty piece. And as regulators, we want to feel really confident when we make a decision. But this whole situation is really uncertain. At some point, those folks had to take a risk to say, we're going to rely on these animal models. So how do we open up that conversation to say, let's allow some of this risk in so that we can make progress?
Well, that's a great point. Go ahead, Jonathan.
Yeah, just to build on what Tina and David were saying, something we think about a lot is, there's this saying by George Box that all models are wrong, but some are useful. And the challenge is, what is ground truth? And ground truth really is when you give a given therapeutic to a group of human beings and you see what happens. Good things happen, bad things happen, maybe a little of both, but more of one. But that's ground truth. What we end up doing as developers in microphysiological systems often is comparing our data to animal model data. Because that's what's available. So we thought, for example, when we were thinking about toxicity, nephrotoxicity, in my case, of a 2x2 factorial design, compounds that were not nephrotoxic in animal models, but were in human beings, things that were toxic in both, or things that are not toxic in either. But it's very hard to fill in that matrix because compounds that were toxic in animal models rarely go into people after that. So you have an empty box there. So you do the best you can to kind of fill it in, but your comparator group is dominantly right now what is, the animal models that people have relied upon for years and years and years to move their therapeutics through the pipeline, the regulatory pipeline. So we have this almost catch-22 situation where the comparator is not ground truth, but the comparator is something that we know often is wrong. I mean, we've cured an awful lot of diseases in animal models that don't translate into cures in human beings. So we know that we have a flawed system now, but when that's the comparator, I think it inhibits the catalyzing these really to be widely adopted until we can get past that roadblock.
Great, thanks. And here, before we open up to the crowd, within the panel, do you have questions for anyone else or across the panel that you'd like to raise?
I'd like to point out too that I think we, in addition to looking at legacy data where often the comparator could be wrong, one can also look about novel readouts. We talk about this session is also about biomarkers. And so some of the things that we have found value with MPS is being able to look at omic technologies. Transcriptomics, for example, we had a study we funded Jamie Thompson and David Page on large language models and regenerative AI back in 2019. And they used purely transcriptomic data from brain organoids to look at developmental neurotoxicity and was able to identify known neurotoxins from just a learning of looking at 100 known developmental neurotoxins and being able to define an algorithm that has a very high sensitivity and specificity. So I think we can also look at those novel readouts to be able to build confidence in the technology.
Yeah, and so, yeah, I think that's an additional topic we could go into in terms of the appropriate readouts as well, within the models, definitely.
Can I just ask something? Very quickly, this is not meant to be a kumbaya statement, but I would love for you, CPAT and complementary to and the regulators to hold vendors responsible, accountable, because I think the key to adoption really is for all of us to exist in this ecosystem where there's a complementarity in which NAMs you use to solve what problem. And instead of sort of competing with each other in this space, if we build a continuum, I think that will easily lead to better adoption across the space.
Definitely. I've had
Yeah.
discussions with Jan about this as well, where some companies' models may better answer a question around specific toxicities for the tubular portion of the kidney, and then look at other portions.
Right.
Definitely. Thanks. So at this point, anything else?
I'll raise one other point that I think is an impediment to widespread industry adoption, and this was at the recent Global MPS Summit. Kimberly Holman, who made a great transition from being a leader academic developer to now being the head of complex in vitro models for Genentech. Her first slide in one of her plenary talks, she said, "When I got to Genentech, I learned that a C57 black mouse costs $2 and I can get 50 different tox readouts from it. A CIVM costs $500 per assay and I get an incomplete readout on a single organ toxicity." So cost is an issue. You really have to show a lot of value added for cost. If there's going to be widespread replacement, 3Rs replacement in industry, I think, of the animal models. I also heard another, the former drug head of safety at Genentech at a meeting years ago at one of our meetings said, I pay attention to cost, but when we're in the middle of a clinical trial and we see a tox problem, and I have to put a halt on 60 plus clinical trial sites around the world, then there's no limit to how much money I will spend to solve that problem as quickly as I can. So I think there are going to be niche uses for these. There'll be, widespread replacement. 3Rs replacement of animal models is going to take a lot of time. Even high throughput screening took a couple decades to be widely adopted in industry. So we shouldn't, have expectations that limit our enthusiasm for continuing the development process in this.
It's definitely, I think, an excellent point that we want to focus on some of the niche areas right now that the models can address. Thanks, Jonathan. And so with that, I'd like to open it up if there's any questions in the audience. Yeah. Could you get a microphone?
Hi. First, I want to say I really appreciate the agency's effort to bringing engineers and drug makers under the same roof. As a mechanical engineer turned to pharmaconutrition, my first job was actually to develop finite elements model for fluid structure interaction models. So when you say you want to challenge in critical path, I would say you need to challenge us, so we're on board. So in light of previous conversation past 2 days, when you talk about VIV40, I think it's worth to bring up VIV70. I think there are two camps of model builders where, you and I, we are based on first principles, physiology, Newtonian mechanics, biochemistry, and there are model builders that are data-centric. How would you like to bring the two camps to a model building and certain quantification?
I think you were listening to our lunch conversation.
Your mic was on?
It must have been on. Actually, Madhu and I were talking. There's an interesting book called The Virtual You, which is a discussion of all the current relevant science to build digital twins. And one of the things that's really lacking from these authors' perspective, as those who are engineers know, the understanding first principles, having theories for how things work really can drive engineering forward, and why in other industries like automotive and aerospace, they only use simulation to get to that first product, right? And we don't in this space. There's good reasons for that. We don't have solid first principle theoretical models to drive all the biology. The complexity is still, from the perspectives again of these authors, is that that's a place to put additional resources. Because when we're building stuff from data-driven models, and he mentioned VNV-70, which is an analogous effort to looking at credibility of AI, which I do not want to open the box today. In order to better drive our observations and the experiments we need to do, if we had better theoretical models, we could target those. But when we're building theories just from data-driven approaches, there are things we can't see. There's massive uncertainties in doing that work. So I think we still have a long way to go in this space to do that. My marks.
Thanks. Any other comments on that question? No. All right, I think we have time for one more question over here.
Yeah, AJ Allen, the Chief Medical Officer for the Institute for Advanced Clinical Trials for Children. Today is sort of about pediatrics with Inc. Presenting and more generally. One of the challenges, and I did lab work some years ago when I was getting my PhD. One of the challenges we have in the pediatric space with, I think, adopting some of these models is development. I mean, just taking the example of a in vitro liver model to look at toxicity, how old is it? Because if you're talking about neonates, there's a lot of changes that occur in the drug metabolizing enzymes in the first few months of life, and that has significant meaning. And so that's one of the things. Now, I don't think necessarily everything has to have a developmental component in that, but you know, it's notorious that a lot of times we don't have models for disease states and things like that in pediatrics because of the difficulty trying to do in vitro models with a temporal component related to development. So, any thoughts on that?
If we've got a few seconds here, if anyone has a comment here around using, pediatric applications.
Sure. Yeah, thank you for the question. So one of the emphasis of our programs within NIH is to look at development, and especially across the lifespan. So one of the cell sources that we heavily use upon are the adult-derived stem cells. And as you know, when you make them pluripotent and differentiate them, they remain into early neonatal or juvenile states. So in some ways, that's we're working with a major pharma was very interested in pediatric applications. So in some ways, that's serendipitous because you can essentially look at the changes in metabolic profile from neonate, juvenile, young adult to adult. And so that's where we're looking at from the same source.
I mean, it seems like that's going to add an additional layer of complexity to validation, right?
Yeah.
Right. Well, thank you everyone for joining us today. I was very pleased to get this diverse group together. So it was wonderful to see all of you today, and I look forward to the rest of today's conference.